gram stain. So let's go look at a couple of these. So gram stain. Open that in a separate tab. There we go. Okay, in the gram stain, let's look at, let's see if we have the next one here to do. Rods. There we go. Here's nice. No, I like that one. There we go. Dark blue. The gram negatives are looking. This is actually one where you're getting both. It's one species of its variability. So this is gram positive. The gram negative is kind of a reddish, pinkish color. Gram negative, right? So the question is going to be, it's going to be a scope. I've done a gram stain. I'm pointing at a cell and I ask, tell me about the cell. It's gram positive, gram negative. I can say, tell me about the cell wall, right? So if it's pointing at this cell, what would you say? What do you know about the cell wall of this organism? Thick peptidoglycan, right? Thin peptidoglycan.
It's gram negative.
This is actually one species, and I don't know why microbiologists aren't honest with students and say that there are species that do change as they get crowded in stationary phase. They start to go gram variable, but you didn't hear me say that.
May I ask you a question?
Yeah.
You're gonna also ask us about the cell structure. Is that what you said?
Yeah.
So we need to know, like, it has this thick peptidoglycan, and then the gram negative has the LPS, is it?
You just went too deep for a lab.
Okay.
So the LPS and things like that, we don't deal with in here. That's something that makes you sick. That's not something in lab that we actually take advantage of at all.
Okay.
Right? That's more of a lecture.
Okay.
Just notice.
Yeah, that's good. I'm glad you asked that because that's because like, what you need to know in here. You need to know things that were important for what we did, and LPS just doesn't turn out to be useful for what we do.
Okay.
If we are infecting mice, yeah, LPS will make them sick as all get out, but we don't do that. That's too deep. So now let's look at the acid fast. There we go. Okay, acid fast. Can you see the difference between this pinkish red and the last one?
Yeah.
That's important. This, I call this boat paint pink. Boat paint pink. I haven't water skied in a long time, and every boat I ever water skied behind on the Colorado River had sort of those sparkly color, shiny, sparkly boat paint, right? That's what they like. So it's, let's go.
Consider it.
Call. Let's go back to gram stain. I'm going to talk between these two.
How do they spray it? Bright. Really, really bright. Really, really bright.
Okay.
It's more pink.
So you have that
It's a little more pink.
Yeah. It really.
versus
Yeah, I was very looking.
that. Right? This washed out pink. If it's washed out, if you're not impressed, you're like, yeah, I can see it, but I'm not going to spend $30,000 to buy a boat that looks like that. That's gram stain. All right. What is the acid fast stain telling you? What is the
Waxy wall.
Hmm?
A waxy cell wall.
Waxy cell wall? Yeah. So remember when you were spreading these, it was like wax. If you didn't spread them out really well, it was kind of like exactly like spreading wax in a liquid. And the book doesn't really go very far on explaining why this is important, but you could think, hey, if you got a waxy cell wall, getting a drug into that bacteria is going to be difficult. Getting anything that is in water solution, aqueous phase, is going to be difficult. one reason why microbacterial infections, microbacterias, that's why they're hard to treat. Yeah. Yeah.
So will you ask a question like what type of bacteria that is?
Sure, why don't we add that? Because it's a simple question. It's either mycobacteria or nocardia. There are two genera in the whole world that will be acid fast. Do I want you to know the genus? Why don't we do it? Because it's an easy one, right? It's either mycobacteria, right, which is a genus, so there's Mycobacterium tuberculosis, Mycobacterium paratuberculosis, Mycobacteria schmegmatis was the one you used, and nocardia. That's the other genus. Only other genus that has acid fast cells. Nocardia.
So if we put one of those parameters for that, it would be right.
Yep. As long as I ask the question, what genus could this be, or what species, or what? Yeah.
But we're gonna have to look at that and then tell.
Yeah.
Would you repeat what you just
Because
said?
that acid fast is only for mycobacteria.
Or nocardia. So the only
Bacteria.
myco.
Myco.
So the only two genera on the planet that we are aware of, genera is the plural of genus, so the only two genera that have acid fast cell walls are Mycobacterium, so that's where you have Mycobacterium tuberculosis, right? So this is a test you would do to see if somebody has tuberculosis, or Nocardia, which is a soil bacteria, which is not, there's people that are immune deficient sometimes get this, but it's very, very rare. The reason you did this is because tuberculosis is a huge problem outside of the United States, right? And it wouldn't be surprising if it came our way in a drug-resistant way. So,
Can you spell that? Nocardia?
Nocardia.
Can you spell mycobacteria?
MYCO bacteria.
Just said it
Yeah.
fast.
Yeah.
So how, if we were on the test work for the practical, were to look at that, maybe if you were to just hypothetically, how would we be able to tell whether it was mycobacteria or nocardia?
You could not. I would never
Oh,
expect
you would
that
just
to
say it would either be.
Yeah, we have a lot of situations
them.
where
Oh, okay.
you're able to rule out a lot of bacteria. Like let's say it grows on an EMB plate and it turns it color, but it doesn't turn it green. Okay, well you can tell me a lot of things, but you cannot definitively identify it from what I've told you. That's fine.
Okay.
Right? The ability to rule out all gram positives or all gram negatives, or in this case, all non-acid fast bacteria is really helpful. That's a good question. Okay. Endospore stain. Only stain that was green. Okay. Let's see if we have endospores in here. Collection, endospore. I should just link to this page, man. Yeah, I will. Okay. Hey, it's going to look something like this. You have green and red. It's the only shade. It looks more green in the microscope. Let's find one that looks more green. There we go. There we go. There you go. All right, tell me about the bacteria that did this. Why'd they do this?
We got a picture screen.
Because of that stain.
Bacteria did it before you stained it. Why did bacteria do this? Right.
They make spores.
Yeah, so in nutrient poor conditions or a lot of cell stress, they form it in this form. Okay? It's like a time capsule. Stick my DNA in this time capsule, it's tough, it's environmentally resistant. These kind of things, you have to autoclave in a special way to get rid of them. They're very tough. In a hospital environment, you're most likely to run into these in terms of Clostridium difficile. Clostridia is a genus that all of them can form endospores. So typically, if you're getting Clostridium difficile, like let's say somebody has it, they are doing what people with Clostridia difficile do, which is fecally shedding. And let's say it's on a surface, and then you pick it up. Right? As much as you don't want to think about it, the fecal oral shedding is the most efficient way to spread. Right? It's disgusting, but it happens a lot. So you touch something, what you have just touched that will transmit it is the spore. The bacteria itself will I'll bet several $100s dies pretty quickly. So any vegetative cells of Clostridia that were on that surface within minutes are dead, and what you are transferring to you is a spore, right? And then it gets to you, germinates in you, and now you are sick. Okay?
So what would you ask us about this particular one?
What test is this? What are the reagents? Right? Alkyl green. Not when we use green. Right? What's the trick? What was the trick in this one?
Standing the spores.
Yeah, but staining. We had to do something a little
You mean
special.
with the heat, the steam?
The heat and steam, right? What cell structures are being targeted? And the spore. Let's see. Okay, evaluation of an osmotic growth. Quite honestly, there's very little I can ask specifically about interpreting these, other than what was the experiment? What was the purpose of the experiment? But I got to be careful there because you only have a little bit of space to do it. That's a very general question. Which tube grew more? I could have A, B, C, and they'll be labeled. Evaluation of media, I will have those 4 media, GSB, ISB. So I'm not going to make it hard for you to just have 4 random tubes and say, you're supposed to know this is a comparison of media. I'll have those on there.
So what was its purpose, you said?
Well, yeah, what's the purpose? You work on it. Okay, osmotic growth, same thing. I'll have 1%, 3%, 11% on the tubes. I wouldn't expect you to know any given tube. What is that? That's not fair, right? But knowing that you have 4 tubes, 1%, 3%, 7%, and 11%, well, then you should be okay. This is comparing ability to grow in salt, that kind of stuff. And I could have 2 rows of tubes. A lot of times what I'll do is I'll have 2 species, A, 1, 3, 7, 11, B, 1, 3, 7, 11, and one will grow at higher salt than the other. And all you have to do is say, oh, A grew better, B grew better.
And then that's where you're going to have the and you're going to have the blank one next to it.
Yeah, I'll always
Okay.
have an uninoculated out so you can compare. This
Okay.
is what uninoculated is.
That's
And
what
the
I
more that it grew, the cloudier it is.
Correct. That's turbidity. We talked about that in lecture. So, yes. Aero tolerance. This is the one where the VHI tube. So we did obligate aerobe, obligate anaerobe, and facultative anaerobe. Those are your 3. Okay. I don't mess around with microaerophile because we never see them in here. They don't do it. Right.
Can you say that again?
Obligate aerobe, obligate anaerobe, facultative anaerobe. That's it.
Those are the only
I don't mess around with the aerotolerant anaerobe because that I know what it means, but I think it muddies. It makes it more turbid in your brain. I think.
Can you just repeat it one more time?
Obligate aerobe, obligate anaerobe, facultative anaerobe. So
Obligate.
basically, the 2 extremes, the one in the middle, facultative anaerobe.
Okay.
Obligate anaerobe does not grow in air.
Yes.
Now it's a little more subtle than that. Remember, in the tube, you're looking for that band where it doesn't grow up top.
Right.
This is the one where you'll need to be uninoculated because you'll need to look at them and go, okay, all right.
I know I have.
Fluid phytoglycolate. I should have taken out the syllabus. We didn't do it. Moving on. Passive agglutination. Our kit's not working, so we didn't do that. Moving on. Soil microbiology, relatively straightforward. You had your Sabarod's plate, which grew the fungi, your glycerol yeast extract, which grew the actinomycetes, and your nutrient agar, whatever, the one that grows just about everything. Okay.
Which one makes it dark yellow? Nutrition agar?
Sabarod is always darker.
Sabarod.
Sabarod's darker.
Okay.
And the fungi look like fungi. Your soil plates are still in there. They're smelling like good soil. If you want to look at the fungi plate, because that one, there'll be one out and they'll be like, which one's the Sabarod plate?
Was that the one with the antibiotic?
Yes. Sometimes with the antibiotic, I don't get one that looks really good. I don't want to ask a question where it's not.
So you're not gonna ask that?
No. Let's say no. It's less for me to do. Sorry. So any questions?
I got lost in those.
The ones that we did not cover that you're not going to test us on were the passive
Passive agglutination,
and the
fluid glycolate.
and what about parasitic protozoans? Didn't you say that was?
We're not gonna.
Yeah, we're skipping that.
And there was one more up on the top. I think it was.
Hand washing. There you go. We did
And
it.
the photosynthetic microbes.
Yeah, photosynthetic microbes. Stay away from.
So,
I can make a test out of it.
So basically,
standard plate count?
Yep. Standard plate count. Doing some there'll be a plate out. It'll like have 4, a certain amount of number of colonies. I'll tell you how it was done. Right? You just got to calculate back the numbers. Essentially, it's just the number, and then however many times, how many zeros you got to add to get back to the total. Isolation streak plates. The only question I can ask is, did it work well? Right? Well, but you should know what you were looking for. So you should be able to look at a plate, and if it looks like this versus this, which worked better? Second one. Yeah, that's about all I could do on that one. It's sort of a technique. What were you looking for, and which one worked better? That's isolation vectors. Those are pretty straightforward. I'll put those in there. Fleas and ticks. Very easy.
Oh, I did that already.
Which one
It's
is?
just identifying.
No, you don't need to use species. You just say tick. And eukaryotic versus bacteria. Know that those aren't bacteria. Know their domain, right? Domain. Eukaryote versus bacteria. Yeah. I will use domain. So I say, what is your domain?
Eukaryotes.
Correct. All right. That's it. Today. Yeah. That's not it. We got some stuff to do. Yes.
On Tuesday, you were saying something about we never got that clear.
Purpose of isolation streak plate is to separate out. You have more than one species in some sort of mix, like those bugs have tons of bacteria in there. I want to separate out one species from all the rest. So what you're doing is you're diluting it out on the plate so that you end up with a single colony. If you have a single colony, that probably came from one or a few cells. By definition, that's one species. Right? And then you can take it off that plate and grow it up completely separate. So let me actually close that loop. Let's see. Back this way. I can show you the purpose of an isolation streak plate. Okay, so I had that bacteria on the insects. What is the purpose? Okay, well, the purpose was I grew up something like this. Right, and this has, how many species are on here?
About 4.
Yeah, more. You can't, I wouldn't expect you to judge anything that's obvious, but yeah, this is probably 4, 3, 4, Right, but that's a mess. I want to study them individually. So what I did and what you'll do when you get your unknown is you get a well-separated colony, and I grew it up in here. So there's the fungus. Now that's the fungus growing up, that species, and there's nothing else in there. Right? I have a pure strain now.
Okay.
And I took a one of the bacteria, and hopefully it's separate. Yeah, looks like I have just that. I don't see any other colony types in there. It looks like I have a pure strain. If I'm lucky, this is something that grows on the inside of the bug, which is interesting. We'll see. That was the purpose.
All right.
Okay.
Thank you.
The purpose is actually
The isolate. and
Isolate.
the program.
Yep.
So with some plays for soil microbiology, what did you want us to go ahead and on there?
Repeat your first part.
What would be an example of something you want us to know from the state?
I'll go on a separate plate. Well, okay. What do I hope you would know? the soil is full of cool stuff. Okay, what is going to be on the test? Saborod's grows fungi, glycerol yeast extract grows actinomycetes. That's kind of limited there. That's a hard one for me to ask many questions on because, you know, I would hope I could put a plate in front of you that had obvious differences like this. Like if you saw a soil microplate, let's say you see it, like, something's up. This is not a soil microplate, but this, what if I did this? Okay, how many species minimum are on this plate?
2.
Yeah, okay, I'm going to put a plate like that on there so everybody got that, right? Right? That's worth rewarding. That's something you've learned this semester, right? That different bacteria, I mean, it's easy, but in a sense, that bacteria, what is heritable, what makes a species, you have an appreciation for it. Why not throw it on there? So I'll put on there and it'll be something similar to that, right?
Appreciate it.
I'll ask for a number though, right? And I'll somehow ask it so that you can't just say more than one, or we'll figure it out.
Can you put it up there one more time?
Yeah.
Okay.
Right. So you can see those little pale colonies and that little fungus that I'm hoping is cordyceps.
Both of the things on there are fungus?
No, this
The white one?
honestly, I think this is a fungus because I can see it's thready. The translucent ones, some fungi can actually be single cell and will grow in colony form and don't form hairs.
So fungus
So I don't know what that is.
are like hairy fuzzy.
All fungi are hairy, but some fungi are single cell, which are called and will grow in colonies. They don't make hairs. So I don't know what the non-hairy one. I don't know what that is. I would need to stain it.
And you wouldn't ask that.
No. That's getting a little too much.
What's that over there? Hold up. What was that?
What? This?
Yeah.
Okay, in notebook. Let's go. Okay, so your Savarot plates are still there. I want a picture of the Savarot auger plate now, because and I have a reason for this. Just put it in your notebook.
For today or
For today. Okay. What else do you have to do? Let's take out this one. Okay. So I want two drawings in the notebook. Okay. The reason I want this is as follows. So trichinella. This is stuff that is not in the book. So you're not expected to know this, but I want you to appreciate what you're actually looking at. So up here we have Trichinella spiralis encysted larva and Trichinella spiralis female WM, which stands for whole mount. I want a drawing of both to scale. And when you draw the circle that you see and draw the organism relative to what you see, so it's the same size. And the reason I'm doing this is Trichinella is something you get typically from eating pigs, although it's in a lot of other species. And what you're actually eating is the encysted larva. The whole goal of this organism is get inside of another organism, encysted in the muscle, and wait for a carnivore. Carnivore eats it, and it goes into the stomach and small intestine, germinates, has sex in us, right, male, female, and then the larva actually crawl through your mucosa, and they will insist in various places. But sometimes they don't come out in the right place. What they're trying to do is get into the muscle, because this is quite clearly an organism that it used to travel and still does in some carnivorous species. It goes sex in someone's stomach, head for the muscle, wait for another carnivore to eat that cyst, go into the gastrointestinal tract, have sex, have babies, larvae go into the muscle, wait for the carnivore, right? That's its life cycle. So all big cats in Africa have their own species of this. Coyotes do, Anywhere where this can travel like this, there is its own species that's in it, and we just happen to have one that primarily we get from eating undercooked pork. How can you protect yourself from this? If you ever see somebody who wants to feed you undercooked pork, insist that they freeze it first. Freeze this, thaw it, it's dead. Okay? So, or just cook pork. So, what you'll be looking at is, I want you to get a sense for the encysted larva, and you'll be looking, you'll see it'll be in muscle. And then the female worm, which is from probably actually a rabbit.
So this is what you want us to draw, the worm and the
Yeah.
it's
So draw the female, which is a worm. Looks no different from any other worm. Yeah, the circle you see, and the encysted larva, which looks a little different. That's why it forms the little spiralis, the spiral thing. Yeah. Yeah, I think I do. I did last semester. Sure do. That's it. That's all I want you to do. So, on the practical, I'm just adding one more parasitic worm. Yep, that's still on there. When you open it up, smell that.
Are we
Yeah.
looking at the
Yes. Yes. Yeah. Mar. Yeah, so sorry. There's three things I want. Picture of Sabro auger. Let me write number 1, 2, 3. Sorry, I started different than I did. 1, 2, 3.
A plain stuff on, like, does this have any growth?
You can't find it.
I already got a fine I gotta do. Sociology. Gotta do that fine. Then hopefully over the weekend. I'm not really confident I'm gonna be prepared for the class.
You don't think so?
About for the final. I mean for the lab practice. I'm gonna study when I need to study. I'm gonna look at that.
Right.
Other notes and stuff.